Hello, everyone. Welcome to today's live broadcast, Transforming High-Performance LC-MS Analysis in Biopharma, from Molecular Characterization to Attribute Monitoring. I'm Laura Bush, the Editorial Director of LCGC, and I'll be your moderator for today's event. We're pleased to bring you this web seminar presented by LCGC and Biopharm International and sponsored by Waters Corporation. With 60 years of research and development of specialty measurement instrumentation, Waters' LC-MS, chemistries, informatics, and thermal analysis innovations help pharmaceutical and biopharmaceutical companies efficiently accelerate and improve workflows. Their deep understanding of the global pharmaceutical industry, combined with their advanced analytical technologies, help bring critical medicines to patients quickly, safely, and effectively. We have a few housekeeping announcements before we start today. First, the webcast is designed to be interactive, so we encourage you to ask questions. You can submit your questions at any time by typing them in the Q&A box, which you can find on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the webcast. And if you have any technical problems viewing or hearing the presentation today, please click on the question mark Help widget in the dock at the bottom of your window. I would now like to introduce today's speakers. We are very pleased to be joined today by Melissa Lee and Scott Berger. Melissa Lee is a principal scientist at Pfizer in the Mass Spectrometry and Biophysical Characterization Group, where she focuses on product and process development using mass spectrometric-based methods. Before joining Pfizer in 2013, she was a postdoctoral research scientist at Agilent Technologies. Melissa Lee received her PhD degree in chemistry from Rensselaer Polytechnic Institute. There, her research focused on the detailed mass spectrometric analysis of complex carbohydrates and biopharmaceutical proteins. She is also an alumna of the University of Vermont, where she received undergraduate degrees in biochemistry and chemistry. Scott J. Berger, a senior manager in the biopharmaceutical markets team at Waters, received his BS from the University of the Sciences in Philadelphia and his PhD in chemistry and biochemistry from the University of Colorado at Boulder. His interest in combining high-resolution separations and mass spectrometry started during his postdoctoral research under Dr. Richard D. Smith at Pacific Northwest National Laboratory, and it continued while he was a principal scientist in the Life Sciences R&D group at Waters. His current technical focus within the Biopharmaceutical Business Group relates to the production of novel and robust separations and LC-MS workflows for the analysis of biotherapeutics. Thank you both for joining us today. Scott, please go ahead and get us started.
Hello, this is Scott Berger from the Waters Biopharm team. It's a pleasure to be here with Melissa today to talk about the broadening of LC-MS technologies within the biopharmaceutical industry and the technology that Waters is producing to help enable that capability. I think it's been a journey in terms of the growing importance of LC-MS for biopharmaceutical development and commercialization. I think one of the signposts in this journey has been a recent paper by the FDA talking about a retrospective analysis of their BLA submissions. So in this paper, they talk about the retrospective analysis of 80 approved BLAs from 2000 to 2015. And the first piece of data they gave us is 79 of these 80s used mass spectrometry for either product characterization or for the characterization of impurities within the product. So really it's become a ubiquitous technology for a commercial release of biotherapeutics. One of the other pieces of data in this paper is looking at the ways mass spectrometry is used, and it's increased over time. The number of molecular attributes that they're looking at. This may be a modification, it may be just simple mass confirmation, it may be an identity test, but the way mass spectrometry is being used has increased over time. The number of aspects of the molecule that they're looking at has increased. When we look at how mass spectrometry has been used, in these BLA applications, according to this paper. It's almost entirely used for characterization of molecules. It's not being used in terms of release assays, at least according to the data they were looking at. But what they did see was that there were three dominant workflows that were being employed for the use of mass spectrometry in these filings. What you see here with these multi-bar charts are over different time periods, the percentage of applications having that workflow incorporated for the characterization, again, of the molecule or of the impurity. So peptide mapping has dominated. Almost every application that has been submitted has had at least some peptide mapping application. Intact mass, very, very close follower in terms of ubiquitous workflow. And then what you see is for glycan profiling, it's probably used in about half of the filings with an MS detection technique. And so their conclusion was that these methods are becoming more established, more accepted, and they expect to see them more broadly throughout BLA applications. The interesting thing was, with all the discussions in the industry about expanding the role of mass spectrometry, they also acknowledged that they expect to see mass spectrometry methodologies expanding within the regulated development world in terms of things like process monitoring, but also expanding into quality control and comparability sections of the applications, even being used as release tests. Able to support that, we really need to expand who can use mass spectrometry today. It's going beyond those core characterization groups. For things like biosimilars, it's going into the discovery groups who may want to do some advanced analytics for clone selection, a key element of selecting the proper clone for a biosimilar product. But it's also going into regulated development, where they typically have not used these technologies directly, and also into QC environments. So expanding the number of scientists who are capable of generating high quality, high information content, MS data, is one of the things that the industry is saying needs to happen in order to really support this broader deployment of LC-MS technologies. Now, it's important that they be able to capture the data, analyze the data, but it's also recognizing that the environments they go into are not these discovery and early development environments, the characterization role, but also into regulatory sensitive environments where compliance-ready tools and informatics are a key requirement. So being able to span that journey from discovery all the way through QC with common platforms. You know, the payoff of all of this, you have more information so you can make faster and better decisions, raising the overall productivity of the commercialization and development efforts. So as we've listened to our customers, as we've had the conversations with them and attended meetings and listened to their concerns about how to really facilitate this expanded access of LC-MS, high information content technologies throughout biopharm, really the number one thing that comes through is the argument of robustness. That if more people are going to be using this system and less experienced people, this system needs to start up quickly and easily, and it needs to stay up and running. And especially in the QC environment, the expectation is for high uptime. What comes in a close second, though, is reproducibility, especially in the regulated environment. You don't want investigations to come from using a technology that is not reproducible. So raising the reproducibility of the techniques was a close second in terms of customer interest. This easy to learn and operate obviously is key for users who are new to mass spectrometry, to get them up and running quickly and to allow them to be effective. But there's also concerns such as limited lab space in environments today, the need for small footprints, and that the informatics, the automation of all of this really needs to flow, and that you need informatics that allow this level of automation, but also handle all the regulatory or process requirements that the companies impose. Our response to all these requests and all the challenges that we're seeing coming from these requests has been the introduction of the BioAccord system. It's a high-resolution TOF-based system for the analysis of biotherapeutic molecules. Featured in this system is a new detector, the Acuity RDA mass detector. It's paired with our Acuity UPLC, the I-class, which is our binary mixing system, so the greatest reproducibility on the separations and the high performance of a compact TOF mass spectrometer. And we'll talk about one that was designed not only for experts but for non-experts to readily acquire data with. Now, depending on the application you're doing, there will be optical based data acquisition on this system, TUV for things like peptide and protein-based workflows, FLR, fluorescence detection for the Hillick glycan analysis. Controlling this in terms of system operation, data processing, and reporting are our Unify Informatics platform. And this system is a holistic system. In fact, it gets tested as a system before it ships from our site and gets tested as a system when it arrives on the customer site. It's tested not for small molecule applications. It's actually tested for large molecule applications, intact mass, peptide mapping, and released glycan analysis as the customer requires. To support this, there are a range of consumables, everything from the installation kit so we can run these applications when the system is put in place in the customer lab, as well as system check standards based on a recognized standard, the NIST monoclonal antibody. So we have an IDE standard, an intact standard, and a tryptic digest standard based on this NIST material, as well as a glycan performance test standard. So with this, we can install the system, show it's working for the application, and train the customer on these workflows. You know, we talk about this as really the first smart MS-enabled biopharm LC-MS solution. It's purposely designed to be a small benchtop system with high performance, to allow more people greater accessibility to this system, to focus on these key protein, peptide, and glycan workflows, and to do so with more automation than people have experienced before, being able to submit a sample and go directly to report or have some facilitated data review as an element of that workflow. It is fully compliance ready. You can have the features off if you're in discovery or development and enable them when you're working with this system in regulated development and QC. So when we talk about Smart MS, really it's the simplicity of interaction with the system. It means that the button you see on the center of the instrument there is the only button on that instrument, and that allows you to start the system up or to shut the system down. When you start the system up, there's an automated setup routine so that you know the system is ready to perform. You can see the green lights on that feedback system. And then an intuitive, simple user interface that tells you if there are any action items required in order to bring the system to full performance. And that intelligent diagnostics is really critical for people who are not mass spec experts. They need to know exactly what part of the system may have an issue and then have an SOP provided with the system on how to resolve those issues. This is an example of startup on the system. So you've turned the system on, and this would be the page where you can do the instrument setup control. It's simply a button, hit start. It will go through the calibration routine. It'll calibrate it both positive and negative mode under all the conditions that you request for calibration. You know, when we talk about intelligent diagnostics on the system, we refer to both that front panel as well as the information behind it, telling you how to resolve any issues with the system. And so you're going to get more feedback than you ever have about the status of your system. Obviously, you hope that it is always in the green, but when it isn't, if there are warning levels or caution levels, you not only get the fact that there is an issue with the system, but all of these icons are meant to indicate to you exactly what part of the system requires attention. So whether it's the fluidics, electronics, maybe the pumps, or communications, all of those have specific icons and specific procedures to resolve any issues you may encounter. I'm going to transition now to the workflows supported by the system. I think Melissa's talk is going to cover this in much more detail in terms of the why and the how. But this was really designed to support those three major workflows in addition to other secondary workflows used for biopharmaceutical analysis, starting with things like intact mass analysis, reduced subunit or IDES digest in the case of monoclonal antibodies or other types of subunit analyses used for other molecules, digestion and peptide mapping, and released glycan analysis based on RapidFluor labeling and analysis. And again, because we're under the Unify architecture, we can acquire, we can process, and we can report under one regulatory compliant system. So we'll start with intact mass and subunit analysis. And I won't belabor this slide too much, but all the tools are delivered with this system to be able to install the system, to be able to test it for this workflow. And those are the same materials that we can use the columns and the standards to educate the users, particularly if they're not familiar with LC-MS workflows, to actually teach them to do the workflow, not just to use the system, but to be effective at doing intact mass analysis. So the data you get out of an LC-MS experiment, an intact mass experiment on a monoclonal antibody such as the NIST-MAB, is a multiply charged envelope, so mass spectrometers separate by mass to charge. So all of these different peaks actually represent the same component with different numbers of charge. We have to apply the Maxent deconvolution algorithm to generate the neutral mass, so generally taking the data from each charge state and putting it into its proper place in the neutral mass space of the deconvoluted spectra. So now we're in the 150 kD measurements of a monoclonal antibody. We can see the different glycoforms that are present on this antibody. Antibodies are glycosylated, so we're seeing that as the dominant effect in this deconvoluted spectra. The relative height of each of these peaks in the Maxent deconvoluted spectra represents the relative amount of each of those glycoforms. Now, it's important that we get a robust, reproducible measurement when we do intact mass, and we can see the result of an experiment looking at over 320 injections of this NIST monoclonal antibody, and we're looking at the important output, which is the relative amount of each of these glycoforms over time across all these injections. And as you can see, we see very low percent RSDs. Even on one of the minor ones, we're seeing less than 5% RSD. Typically, we're seeing less than 2% RSD on each of these glycoforms. When we're doing intact mass analysis, we have the flexibility on this system to do both native and denatured intact mass analysis. The mass to charge range on this system is 7,000 in positive mode, and there is the flexibility of using your existing methods with TFA or formic acid as the acidic modifier. You know, it's just as important, especially in the regulated environment, that if you have labs at different sites or multiple instrument on one site, that you're getting the same results. So we did this same analysis 8 different times on 6 different systems, actually at multiple locations, and the results are presented here, where we're getting the same results and consistent results over those 6 injections over 6 different sites. For peptide mapping and monitoring, again, we have the standards, we have the columns, and we have the informatic workflows to support both installation testing and training of a customer. And I think one of the areas in which this system will be used extensively is this monitoring application, in which people are looking to take the attributes that they've discovered during characterization and monitor them later in development and potentially for QC. And we support that workflow as well. So if we think about characterization, yes, the system is capable of doing some basic characterization work. We can get high coverage. We have the ability, even though it's a TOF-based instrument, there is no quadrupole, so it's not a QTOF-based instrument, but we can do a high-low energy state post-source, collect both those data sets at the same time, so we can have fragmentation for everything in the sample. When we're thinking of monitoring, we're going to choose specific peptides. We're going to target those for analysis by their mass and retention. It may be something like a modification state of a given peptide, so we would choose the native and the oxidized form. We would monitor those two peaks, quantify those two peaks, and calculate percent oxidation. So here we've done this for the Miser peptide. It's a typical high oxidation peptide on a monoclonal antibody. We're seeing it at about 2.5% within that sample, and we can see an RSD over a large number of injections of about 2%. Released glycan is the third workflow supported by this system. And with this, it's a more complex labeling workflow, but we've worked very hard with our Rapiflora MS technology and the kits that come with it to simplify that, both in terms of the number of steps, making success much greater for users of this system than typical reagents, as well as dramatically reducing the amount of time it takes to get these labeled samples. When these samples are labeled, they have higher optical fluorescence absorptivity, so more sensitivity for fluorescence quantification, as well as extremely good MS sensitivity compared to the traditional reagents used for glycan analysis. So in this workflow, the glycan installation kit, people would be trained to do the glycan analytical workflow, including the use of the Rapiflora MS database that we have within Unify to help automated assignment of the peaks from these analysis. So this would be a typical released glycan analysis. And with our performance test standards, so you can see the various peaks being recognized in fluorescence. Each of those will have high-resolution MS mass confirmation. And you can see, due to the use of the Rapiflora MS label, even something on the lower right, a little 1% abundance glycan peak will produce a good XIC as well as a very good MS spectrum for mass confirmation. So by using the Rapiflora MS technology and the automation of this workflow, we can go from preparing a sample much easier than traditionally done, injecting that sample, and looking at the results very quickly and efficiently. So, of course, all of this needs to be communicated out, and the ability to generate automated reports is a critical aspect of the system. There's an excellent report generator. We supply default templates for people to start out with. And whether you're doing intact, peptide mapping, or released glycan analysis, we can produce all the experimental conditions, the optical data, the mass spectrometry data, and the summarized tables, even including custom calculations within these reports that are required for a customer to be able to communicate out their results. This holistic system, again, very easy to start up, easy to maintain, streamlined workflows, being able to diagnose and resolve issues quickly, and being able to put this in a non-regulated or regulated laboratory is really the foundational architecture that we've built with this BioAccord system. I'm very happy to turn this over to Melissa to talk about results that she and her colleagues have gotten using this system. Melissa?
Thanks, Scott. Hi, thanks for the opportunity to speak today about this high-resolution, small-footprint LC-MS system and its applications for characterizing and routine monitoring of quality attributes in monoclonal antibodies. Especially thank you to Waters for inviting us to visit them. We were there for about a week to do a demonstration on their new system using our samples and methods, and I'll be showing that data today. I'm part of a mass spectrometry and biophysical characterization group, or MSBC, which is part of analytical R&D. They're where we specifically support process and product development. To let you know the outline first, I'll introduce product quality attributes and then go over our platform methods that we use to benchmark the instrument. So product quality attributes can be non-critical or critical. A critical quality attribute, or CQA, is a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. From a regulatory perspective, critical quality attributes of products directly affect the clinical outcome or patient experiences. Therefore, CQAs define product quality and are controlled by process parameters and analytical testing and specifications. a product quality attribute that imparts a change in efficacy or potency, safety or immunogenicity or pharmacokinetics may be classified as a critical quality attribute. And the criticality of product quality attributes are assigned via matrix: severity versus uncertainty. In our group, we characterize a lot of mAbs. The goal of our characterization is to have a well-characterized biotherapeutic. For an IgG1, here is a general list of the many product quality attributes that exist. There are many PQAs that we can detect and follow through process and product development. And in our group, we detect these using liquid chromatography, mass spectrometry-based analyses. And for this testing, mass spectrometry is a central tool for PQA characterization. And for this talk, we are going to be focused on specific product quality attributes that are highlighted in red here, such as molecular mass and size, proteoforms, primary structure and post-translational modifications, and linked oligosaccharides, and also stability or degradation products. And as I said earlier, for this PQA routine testing, it would be highly beneficial to have a high-resolution mass spectrometry for high-quality data. The instrument we tested with Waters is their new integrated BioAccord high-resolution LC-MS system. It is configured as a complete LC-MS system, as Scott mentioned, and it has a very small footprint. It is integrated with the UPLC, and it is easy to use and should be intuitive, like an optical detector. Also, it has easier maintenance and pre-optimized configuration with minimal tuning for the user. The system is also end-to-end in that it uses one software for calibration, data acquisition, analysis, investigation, and reporting. This system, as mentioned, is called the BioAccord system, and the mass spectrometry detector is named the Acuity RDA mass detector. I'm going to call the whole thing the BioAccord system. So going into this evaluation, we had an understanding that the system has a small footprint and that the specification is very different from our research-grade QTOF. And we were excited to see how the BioAccord system fares with our research-grade QTOF when we tested our well-established platform methods on the BioAccord. And these are our four platform methods for heightened characterization by mass spectrometry. This includes analyses such as intact mass, subunit, peptide mapping, and glycan profiling. So from intact mass, we can observe molecular mass determinations that provide information about primary structure, product proteoform heterogeneity, and also multi-chain architecture. From subunit analysis, we can also see PTM and proteoform heterogeneity. That's the IDES digestion with reduction. And for peptide mapping, we can also confirm our PTM and provide a site-specific elucidation of the post-translational modification. And we can determine expected and unexpected and linked glycan structures and abundances. Here are some example either mass spectra or chromatogram results as specified, such as size exclusion, chromatography coupled to UV, and mass detection. And so for this talk, data was obtained for each of these platform methods, and we have a small case study for each. We analyzed 3 different antibodies to observe these PQA examples. So for intact mass, we're going to observe proteoforms, glycation, trisulfides. Subunit, we're going to look at a sequence variant. And for peptide mapping, we'll see some product quality attributes such as deamidation, oxidation, these termini, and we'll look at some N glycans as well. Now we're going to get into the data. Here we see a clean raw mass spectrum of the intact antibody, and it has a good signal to noise, and this is MAB1. And this instrument collects a wider mass range for intact MAB analysis. And on the top panel is the mass spectrum containing the charge state envelope from charge state 30 to upwards of about charge state 70 of the intact MAB. And on the bottom panel, it has been treated with PNGSF for a DN glycosylation to reduce heterogeneity caused by N glycoform. The raw mass spectrum is then deconvoluted with MECTA1 deconvolution algorithm, and then we see the deconvoluted mass spectrum here for MAB1, and we can observe the major and trace level species. So we can see on the top panel the main species is G0S, and on the bottom we see the D-N-glycosylated MAB, and our mass error is very good. And this is with the on the slide background subtraction or IDC on. So now on the top panel is data from our research grade instrument, which is the same MAB1. So the mass errors of both instruments here are within 10 ppm as expected. And overall, there's a similar species and mass profile observed. Keep in mind that the deconvolution algorithm used by the different softwares are not the same. So we expect the antibody profile here to be a bit different. The top deconvolution is a better representation of the raw spectra, raw data, and this data was collected with IDC for the BioAccord. One example that we follow product quality attributes are trisulfide bonds. And that's going to be our first case study. So a trisulfide bond is a disulfide linkage with an additional sulfur atom. Trisulfides are actually a common modification in antibodies, IgGs, occurring predominantly between the light chain and heavy chain for an IgG1. The root cause of trisulfides is related to cell culture conditions. And we have two methods we use for trisulfide quantitation and detection. This includes the intact MAB analysis and non-reduced peptide mapping for a sensitive site-specific method. So this is MAB2, our second antibody, which has a known level of trisulfide. We observe the mass addition here of plus 32 Daltons and use the relative peak height to determine the trisulfide level to be 29%, which is actually the same relative abundance we observe on our research grade instrument. Again, there's very good mass accuracy here. Moving on to subunit, here we have a 3-part subunit, all of which are about 25 kilodaltons separated on reverse phase C4 chromatography, and here we observe the single chain FC, the light chain, and the FD prime. And on the top panel is our platform method, and on the bottom is the BioAccord. So underneath each peak, this is the raw spectra for each subunit. So we have single chain FC, light chain, and FD prime. And we see here very clean signal to noise. And then we deconvolute the spectra again to see the deconvoluted species and average mass. And we see the subunits as their expected species. From the top panel, we see the 0F as the N-glycoform for the major species, and the light chain and the FD prime are unmodified. We observe really low levels of modifications such as glycation, C-terminal lysine, and amidated proline as well, which were expected. And the observed subunit masses match really well with the theoretical masses. So for the subunit assay, our case study for this method is the detection of sequence variants. Sequence variants are unintended amino acid substitutions or sequence variants that may be present at low levels and can arise from several causes, most likely mutations in genomic DNA or mistranslation or misreading of codons or a mischarged tRNA. And that's known as the misincorporation. And all of this might occur during a CHO cell protein expression. And for biotherapeutics, detection of sequence variants early in development may aid in their elimination and reduce product heterogeneity. So now we are back to the subunit assay that I showed before. So there was a threonine to isoleucine sequence variant that was previously detected at trace levels by LCMS. And so initially, this was detected by a combination of LCMS subunit and LCMS-MS sequence variant analysis at a level of about 0.3%. So the sequence variant actually has a known retention time in mass by LCMS subunit analysis. And so we were able to observe this. using our data from the BioAccord. And so we found that the BioAccord system has sufficient resolution and sensitivity to confirm chromatographically separated plus 12 Dalton mass addition at a level of about 0.3%. So on the left, we have the full zoom view, and we have the single chain Fc, we have the aglycosylated single chain Fc, and then we have the sequence variant. And we can see here there's a very small peak for the plus 12 Dalton sequence variant, which was detected in this data. Next, we have N-glycan. So this is a release 2AB labeled N-glycan assay result, and this is a MAB3. And so chromatographically, by fluorescence detection, we detected a very similar profile of N-linked oligosaccharides between our platform analysis and the results from the BioAccord. Now for each peak, we have an example of a major, minor, and trace level species of N-linked glycan, N-linked oligosaccharide glycan, and we have a mass spectra for it. It looks very clean with good signal to noise. Okay, so finally, for the last part of the talk, I'll go over peptide mapping. I'm comparing product quality attribute characterization versus routine monitoring. So for high-end characterization, this includes LC-MSMS peptide mapping, and that involves expert mass spectrometry users with a high level of prior knowledge and literature using research-grade mass spectrometry instruments to determine, discover originally unknown site-specific modifications and identifications and characterize them. MAM, or multi-attribute method, is currently a hot topic in biotherapeutics, and this is a trend to use LC-MS peptide mapping for routine monitoring. In the multi-attribute method, or MAM, routine monitoring is performed with known product quality attributes, and these are targeted search is performed based on the mass and relative abundance is quantitated using peak area from the chromatogram. MAM ideally should involve using simplified instrumentation such as the BioAccord, and new peak detection should be automated. So these are some typical PQAs that are detected and followed through process and product development. PQAs such as heavy chain CDR deamidation or deamidation of the pennypeptide, and also isomerization or fragmentation. And these are their respective release methods. So fragmentation, we would use reducing CGE. The release method for glycosylation is Hillick fluorescence detection. And charge-based separations are also used to study deamidation. And last, for the heavy chain oxidation, we have a focused peptide mapping. And using MAM, it is thought that these PQAs, which are typically using release methods, can be studied using one LC-MS peptide map. And so MAM also allows the additional monitoring of other modifications which may not be detected by our release methods, and these include modifications such as aglycosylation, O-glycosylation, secondary or tertiary oligosaccharide sites. This also includes modifications such as methionine and tryptophan oxidations, and or C-terminal isoforms, and modifications such as glycation. Now for peptide mapping with the Unify software. This is MAB1, shown previously in intact and subunit assays. And this was digested using a low artifact trypsin digest procedure in which the MAB was denatured, alkylated, buffer exchanged, and digested with minimal time and temperature to reduce method-related artifacts. These are the chromatograms from the BioAccord system. On the top panel is a control, and on the bottom is a light-exposed sample. It was light-exposed for 11 days. And we will also look at thermal stress later. And minor changes in the UV chromatogram are starred due to oxidation. This instrument is actually capable of some MSMS fragmentation. This example characterization is done during LC-MSMS, and that assigns the plus 16 Dalton shift to methionine 249 to look at the site-specific oxidation of the methionine oxidation in the FC region. So we can see from the MSMS spectra that the Y4, Y5, and Y6 have a mass addition of plus 16 Dalton, indicating a site-specific mass addition to methionine 249. So routine monitoring is performed using LC-MS, And these peptides in their oxidized and unmodified forms were searched for in the peptide map. So on the y-axis, we have the percent abundance of the oxidation that's occurring. And for the x-axis, we have where the relative abundance is coming from, either the released LCUB peptide map or data from an Exactive Plus. This is data performed by our MAM team, or data that was from the BioAccord demonstration. And so the purple bars are time 0 and the blue bars are t equals 11D. And so these peptides and their oxidized form were searched for in the peptide map. And we can see that the FC methionine 249 oxidation levels for T0 and the stress sample are very comparable between the three methods. The BioAccord, along with the MAM team results, provide additional information and new capabilities beyond the release assay, since we can do a site-specific monitoring of methionine and tryptophan oxidation levels throughout the molecule. So previously we were looking at photostability very briefly, and now we're looking at the data that's coming from the thermal stability. On the left of the slide are imaged capillary electrophoresis results. So we have the T0 on the left and the T equals 28 days on the right. We initially evaluated the charge heterogeneity of the acidic and basic forms of the thermally stressed materials. And we see an increase in our acidic species while the basic species remains unchanged after thermal stress. And so PQAs that might be contributing to these acidic species include heavy chain CDR deamidation, penny-K peptide deamidation, or VSNK deamidation in the FC region. Contributions to the basic species may include C-terminal heterogeneity. So this is looking at the C-terminal heterogeneity of MAB1. On the top panel here, we have the extracted ion chromatograms of the C-terminus in their different forms, either unmodified with lysine or with amidated proline. And underneath those chromatograms, we have the mass spectra for each peptide, showing a very good mass error. And we see that for these thermally stressed samples, for the basic species, the change is due to similar levels, or the lack of change is due to very similar levels of amidated proline and lysine at the C-terminus, which was unaffected by the thermal stress. So continuing to look at thermal stability, here we are looking specifically at asparagine deamidation. And on the left, we are looking at the ICE profile, and we see an increase in the acidic species. And on the right, we have the relative abundance of deamidation of the penny-K peptide. And we see that the penny-K peptide is increasing in deamidation after thermal stress, as expected. And actually, VSNK deamidation is actually the major cause of the increased acidic ice species, which was determined by the MAM team. It actually required a different experiment since the tryptic peptide is very small and not retained on our column. So wrapping it up, this is our summary of the demonstration of this new instrument. And we observed from our methods and experiments that the small footprint BioAccord generated excellent mass spectral data quality. And we found that the resolution, mass accuracy, and sensitivity were acceptable for all analyses. Platform LCMS methods worked well on this instrument as well. And we had done INTACT, where we identified N-glycoforms, trisulfide glycation. We did a subunit where we observed product isoforms and also revealed sequence variants. And we also looked at release 2AB N-glycans and observed the same N-glycans as previously determined. And we did a trypsin peptide mapping to determine site-specific PKAs and levels of modifications such as C-terminal heterogeneity or tryptophan and methionine oxidation and penny-K deamidation using Unify for some thermally and light exposed stability samples. So overall, we found that this BioAccord system has the potential to robustly monitor PQAs for biotherapeutics, especially due to its simplified operation and data analysis, improving user accessibility. Future applications for the BioAccord LCMS system might include post-translation modification or hotspot analysis of the CDRs, structure function studies, process development, and product stability studies. Overall, routine LC-MS will increase process and product knowledge. And I just want to acknowledge and thank everyone that contributed to this data set and presentation, especially at Waters and Pfizer.